So the issue with the regulations that are in place and many of the guidances is they're written in large part coming out of the small molecule arena with perhaps some adaptations for biologics, and more recently, especially under FDA Commissioner Scott Gottlieb, has been very forward-looking and thinking about how can we integrate these new gene-modified cell technologies and manage expectations in terms of quality with what we're able to work with in a biologic system. So for example, we have a dividing drug. What's in the cryopreserved bag that is administered to the patient is not really the final product. It's how that behaves in the patient, how it expands, how it sees tumor, how it's able to kill tumor and leukemia and lymphoma. So that's not really something that we can definitively measure when we cryopreserve those cells. Can we look back in that product? What are the correlative biomarkers that can predict some quality, some consistency in the cell product? Additionally, we look at the gene modification, testing for replication-competent lentivirus or replication-competent lentivirus. There are assays that take some time to perform, and there has not, to date, in several thousand patients that have been treated, been observed a replication-competent lentivirus. So how can we take that back and look at adjustments to the guidances in lieu of what we know about the relative risk? So all of these things are not in steady state. They are hopefully advancing so that we can integrate the latest analytics and the latest knowledge on risk-benefit into the appropriate regulatory framework.